Hi, welcome to my presentation on Chapter 9, Cellular Respiration and Fermentation. So cellular respiration and fermentation are some really important processes in metabolism. Metabolism is just all of the chemical reactions in the cell. So technically, any chemical reaction that we've mentioned is part of metabolism. Any enzyme is catalyzing a chemical reaction, so all enzymes are contributing to metabolism. There's two basic types of metabolism, catabolism and anabolism. In catabolism, you are taking larger molecules and breaking them down into smaller or simpler molecules. In anabolism, you're taking smaller molecules or simpler molecules and putting them together to make larger and more complicated molecules. A lot of the time when we use the word metabolism, what we're actually talking about is catabolic reactions, where you are taking in larger molecules and breaking them down. In catabolism, you're actually producing energy as well as heat. And some of the energy that's produced in catabolism is going to be stored as ATP. So a lot of the time when we talk about metabolism, we actually mean those catabolic reactions that are used to make ATP. But technically all reactions in the cell are a part of metabolism. In anabolism, you're making larger molecules, and that actually requires energy. So you're producing energy in catabolism that's going to be used in anabolism to fuel the anabolic reactions to make more complicated molecules. And in anabolism, you're also going to produce some heat, just because anytime that you have a chemical reaction, you're going to have some energy transferred inefficiently, and some of it is going to end up as heat energy. We have a lot of pathways in metabolism. A metabolic pathway is a series of reactions that always occur together in a certain order, or it's a chain of reactions that is producing some overall product. So in the processes that we talk about today, cellular respiration and fermentation, those all include metabolic pathways. So it's not just a single reaction, but it's a bunch. It's a sequence of many reactions that all happen in a certain order to produce the overall product. The basic purpose of metabolism for organisms is to serve growth and reproduction. In order to grow bigger, organisms need to make more complicated molecules. They need to make their macromolecules. That requires energy, so then they need to take in nutrients and break them down in catabolism to get ATP out of them, or to get energy out of them and use it to make ATP. Then they use that ATP in anabolism to build macromolecules, to make new structures for cells and to make new cells, and ultimately to make a new organism. Metabolism is also used to maintain homeostasis. Homeostasis are your basic stable conditions in your body that you have to survive. So that includes like maintaining your body temperature at a stable level, maintaining the pH of your blood at a stable level. It includes a lot of different things actually. So metabolism is allowing you to do these two kind of things, growth and reproduction and homeostasis, and that means that it is essential for life. The image here is actually a summary of all known metabolic reactions in the cell. This was available as a poster, so like you could buy this as a poster. Not anymore, but you used to be able to from Roche, and it would be like a giant poster that would basically take up a whole wall. So you obviously can't make out a lot of details in here, but all these lines are representing different metabolic pathways. You can see some of them are more like cycles, where they're circles or spirals. It's very, very complicated, and that's basically the only thing to take away from it is just that it's really complicated. So metabolism is huge. There's many, many, many metabolic pathways in the cell. We're going to cover a few of them today for cellular respiration and for fermentation. We're not even going to cover them in super detail, but even so, they're still quite complicated. Like, we're not going to go over like every single step in the pathway, just we're going to talk about the overall things that are produced by each pathway, and even that is still going to be complicated enough for us. So it's complicated. So these two processes that we're going to talk about in this chapter, cellular respiration and fermentation, are the two major ways that cells make ATP from glucose. Whenever cells make ATP, they're never making it to kind of keep it around. You can't really store ATP. So they're going to make it and they're going to use it. And that means that they're continuously needing to make more ATP. They can't ever store it. You can store glucose, when you're storing glucose, you would actually store it as either starch or glycogen, but you can't store ATP. In order to make ATP, you always have to add a phosphate group to ADP. So you're always starting with ADP that has 2 phosphates on it and adding one more phosphate group to get ATP, which has 3 phosphates. Then when you use the ATP, you just take off that last phosphate group and you get back ADP. So you have this kind of pool of ADP that's cycling back and forth between ATP and ADP. Adding that extra phosphate group to ADP to get back to ATP costs a lot of energy. Most organisms are going to use glucose as a source for that energy, although most organisms will also be able to use like multiple things. So they could use other sugars, not just glucose. They could also use proteins or fats. Different organisms will have kind of a different profile of molecules that they're able to use to make ATP. There's actually some organisms that can use like inorganic chemicals like sulfur compounds, but we're not going to worry about them. If you're using glucose to make ATP, then you're either doing it by fermentation or by cellular respiration. Those are both kind of larger processes. Particularly cellular respiration is a very big process. It has multiple metabolic pathways within it, and some of the intermediates in those pathways can also be used for anabolic reactions as well. So that means that the processes of cellular respiration, which are catabolic pathways, are also intimately connected to anabolic pathways that the cell would use to build things instead of breaking them down. This diagram here is kind of giving an overview of how energy travels in organisms. So you're always starting out with plants that are making energy by photosynthesis. So they're actually going to use carbon dioxide and water and sunlight to produce sugars. That also produces ATP, of course, but actually the main thing they're making in this process is the sugars. So that's your basic source for carbohydrates. So they're going to use those carbohydrates to build all of the structures in the cell that plants require, and then you have an animal come along and eat those plants and get the carbohydrates out of it. And that's your source for carbohydrates. Carbohydrates can be, of course, they're going to include glucose, but they can also be converted to glucose. Glucose is used as a source for making ATP. If you have extra glucose, you can also store it as glycogen. If you're a plant, then you would be storing it as starch instead. You can also use fats to store energy. Fats are, as you remember, they're consisting of 3 fatty acids and a glycerol molecule. That glycerol molecule could actually be converted to glucose, and glucose could be converted to glycerol. So you can actually store glucose as fat in a way if you have extra fatty acids to add on to it. So those are the ways that you would be storing glucose to use for later. Then you have your pool of glucose that you're kind of trying to use right now to get energy that you can use, to do some stuff. So in order to do that, you need to basically use this glucose to make ATP, and you have 2 processes for doing that in most cells: cellular respiration and fermentation. In cellular respiration, you're using your glucose, you're also using oxygen, and of course your ADP and a free phosphate, and you're producing carbon dioxide, water, and ATP. In fermentation, you're using glucose, ADP, and a free phosphate, and you're producing some type of small organic molecule and ATP. So you can see one major difference between cellular respiration and glucose is that cellular respiration and fermentation is that cellular respiration requires oxygen. Fermentation does not require oxygen. Another difference is in the products. Cellular respiration produces carbon dioxide and water in addition to producing ATP. Fermentation is going to produce small organic molecules. So that could be stuff like alcohol, which is ethanol, or it could be like lactate, There's a bunch of different things that can be produced through fermentation. So that actually kind of means that there's multiple types of fermentation. Different cells will have the ability to do different types of fermentation. So some organisms can do a fermentation where they make alcohol as a result. Other organisms, like our own cells, actually are able to do a type of fermentation that produces lactate. And there's also a bunch of other things that could be produced in other types of fermentation. So here you have a basic comparison of cellular respiration and fermentation. So first, cellular respiration fully oxidizes all of the carbons in glucose to carbon dioxide. Carbon dioxide, sorry. So all of the carbons that you start with in your glucose molecule are going to end up as part of a carbon dioxide molecule, bonded to two oxygens. Whenever you have carbon bonded to oxygen, that's a very polar bond because oxygen is a lot more electronegative than carbon is. So that means that the electrons are not equally shared in the bond. They're actually going to be closer to the oxygen and further away from the carbon. So when you have carbon and glucose, it's mostly bonded to hydrogens and to other carbons. Those are non-polar bonds in which the electrons are shared equally. Then those carbons will end up having their electrons kind of mostly, partially stolen from them by oxygen in carbon dioxide. So that means that carbon starts out with electrons that are closer to it when it's in glucose, and it ends with electrons that are further away from it when it's in carbon dioxide. So we say that that's like the carbon is losing electrons or being oxidized. So in cellular respiration, the carbons of glucose are oxidized to carbon dioxide. In fermentation, you do not fully oxidize all of the carbons in glucose. So you are, all the carbons in glucose that you start out with that are in nonpolar bonds, they're going to end up as part of whatever organic molecule is being produced by this fermentation reaction. Maybe it's ethanol, maybe it's lactate. Another name for lactate is lactic acid. So different organic molecules that they can end up as a part of. When you have carbons in these little organic molecules that are produced from the fermentation reactions, some of those carbons are going to be part of a nonpolar bond. They're going to be bonded to another carbon or to a hydrogen or something. Some of those carbons will be in polar bonds with oxygen. So some of the carbons have been oxidized, and some of them have not. The carbons that end up in a polar bond with oxygen have been oxidized. The carbons that end up in a nonpolar bond with another carbon or with a hydrogen are not oxidized. So in cellular respiration, every carbon ends up in a polar bond with oxygen, so every carbon is oxidized. In fermentation, only some of the carbons end up in a polar bond with oxygen, so only some of the carbons are oxidized. Another difference is that cellular respiration makes a lot of ATP and fermentation only makes a little bit. That is partly because the carbons in glucose are not fully oxidized by fermentation. When you oxidize the carbons, you're kind of removing energy from them. So if you don't oxidize all of them, then you're leaving some energy behind in these little organic molecules that are produced by the fermentation reaction. That means you're going to make less ATP overall. Also, cellular respiration needs oxygen. Fermentation does not need oxygen. Technically, there's two types of cellular respiration. There's aerobic cellular respiration, which is the main one we're going to talk about, and there's also anaerobic cellular respiration. Only the aerobic form needs oxygen. There's also the anaerobic form that does not, but we'll get a little bit more into that later on. We're mostly going to focus on aerobic cellular respiration, which does need oxygen and is the type that your own cells are able to do. Cellular respiration mostly happens in mitochondria. It also has some reactions that occur in the cytosol, but mostly it's going on in the mitochondria. Fermentation just happens in the cytosol. Cellular respiration is a much bigger, a longer, more complicated process. It has multiple metabolic pathways within it. Fermentation is much shorter. It does still have a couple of metabolic pathways in it, but they're shorter, simpler. Overall, it's more simpler. And then you have your overall equations. So for cellular respiration, the overall equation is 1 glucose and 6 oxygens are the reactants. and they react to form 6 carbon dioxides, 6 waters, and energy in the form of ATP. Fermentation, the overall equation is going to depend on what type of fermentation is going on, and there's a lot of different types, but the one that your own cells are able to do is the one that produces lactic acid. The equation for that is that you start with 1 glucose as a product, as a reactant, and then your products are 2 lactic acids and energy as ATP. So these are the basic processes or metabolic pathways that occur in cellular respiration and in fermentation. In cellular respiration, you have 4 of them. In fermentation, you have 2. In either case, whether it's cellular respiration or if it's fermentation, you always start with glycolysis. Then in cellular respiration, after glycolysis, you do pyruvate oxidation. After pyruvate oxidation, you do the citric acid cycle, and then finally you finish with oxidative phosphorylation. In fermentation, after you do glycolysis, you move on to your fermentation reaction. And there's different types of fermentation reactions. Each one has its own name, but there's so many different types. Different cells would be able to do a different type of fermentation reaction. So we'll just leave it kind of vague as fermentation. For some of your processes in cellular respiration, they have multiple names. For pyruvate oxidation, it can also be called pyruvate processing, or the bridge stage, or the bridge step. The citric acid cycle can also be called Krebs cycle, or the tricarboxylic acid cycle. If it's being called the citric acid cycle, then it's abbreviated as CAC. If it's called the tricarboxylic acid cycle, then it's abbreviated as TCA. I like to call it the Krebs cycle because then I could just say Krebs and it's only 1 syllable. Then for oxidative phosphorylation, that's just called oxidative phosphorylation, but it actually does have 2 processes kind of hiding within it. The first is the electron transport chain, and the second is chemiosmosis. This diagram is kind of summarizing the different processes in respiration and fermentation. You always start with glycolysis. Then if you're doing fermentation after glycolysis, you do your fermentation reaction, whatever it may be. If you're doing respiration, then after you're doing glycolysis, you do pyruvate oxidation. That is actually not shown in this diagram because it's really, really short actually. But it would be occurring right here where you have a carbon dioxide coming out. That's where you have pyruvate oxidation happening. Then after that, you have the Krebs cycle, also known as the citric acid cycle or the tricarboxylic cycle. And after that, you have oxidative phosphorylation. And again, they have just shown that as the 2 separate processes. So first you have the electron transport chain right here, actually including this little bit too. And then on the bottom where it actually shows ATP getting made, that is chemiosmosis. So we're going to go through all of these processes in more detail. This diagram is actually kind of showing in general what is created by these processes, but we're going to go through it one by one. Before we get into the details of these processes though, I just wanted to refresh your memory on electron carriers. Electron carriers are molecules that carry electrons around in the cell from one point to another, and they play a major role in both cellular respiration and in fermentation. There's 3 major electron carriers that are found in cells. You have NAD+, NADP+, and FAD. And for each of those, that's the form that does not have electrons on it. So for NAD+, when you add electrons to it, then it becomes NADH. For NADP+, when you add electrons, it becomes NADPH. And for FAD, when you add electrons, it becomes FADH2. It's the NAD+/NADH and the FAD/FADH2 that are playing roles in cellular respiration. Both of them play a role in cellular respiration, and then NAD+/NADH also plays a role in fermentation. The NADP+/NADPH is mostly relevant in plants for photosynthesis. The figure over here is showing one of those electron carriers. On the top is actually the entire molecule. This is NAD+. So it's the form that does not have electrons on it. and the part that is circled in red is the part of the molecule that actually carries the electrons. So the bottom figure is zoomed in on that part of the NAD+ molecule that actually carries the electrons. The rib here stands for ribose, so that's this part here, and then the ADP is all of this. So this is kind of almost like an abbreviation for the rest of the molecule, and then here's that part that is circled in red where the electrons are actually attached. So this is NAD+, which does not have electrons on it. If you were to reduce it by adding electrons to it, then it would become this, basically. You've essentially added an electron to this nitrogen so that it loses its positive charge, and you have added a hydrogen across the ring from the nitrogen, and of course a hydrogen is just a proton and an electron, so anytime you add a hydrogen to something, you're also adding electrons to it. So when those electron carriers like NADH have electrons on them, then they are considered reduced because they've had electrons added to them. When you remove electrons from them, then you are oxidizing them. So when you go from NADH to NAD+, that's oxidation. When you go from NAD+ to NADH, that's reduction. So now we'll go through the processes in cellular respiration, and we'll cover fermentation at the end. So here you have a basic overview of the processes that occur in cellular respiration. You start with glycolysis. Next you have pyruvate oxidation, also called pyruvate processing. Then you have the Krebs cycle, also called the citric acid cycle. Finally, you have oxidative phosphorylation, which includes the electron transport chain and chemiosmosis. So you start out with glucose. That goes into glycolysis and is converted to pyruvate. Pyruvate enters pyruvate oxidation and is converted to acetyl-CoA. Acetyl-CoA enters the citric acid cycle or Krebs cycle and is converted to carbon dioxide. Meanwhile, as these processes are occurring, you are adding electrons to NAD+ to give NADH. And in the Krebs cycle, you also add electrons to FAD to give you FADH2. Then all of these electron carriers are feeding into oxidative phosphorylation and the electron transport chain. And that is where you're getting most of your ATP. We will go through each of these processes individually. So the first process that occurs in cellular respiration is glycolysis. This happens in the cytosol for both prokaryotes and for eukaryotes. There's a total of 10 different reactions that occur in glycolysis, so it's its own metabolic pathway with 10 different steps in it. Each of those reactions has a specific enzyme that catalyzes it, so you have 10 different enzymes that are involved in making glycolysis run. The overall equation for glycolysis, the overall chemical equation is on the reactant side you have glucose, 2 ATPs, 4 ADPs, and 2 NAD+, and on the product side 2 pyruvates, 2 ADPs, 4 ATPs, and 2 NADHs. So what's actually happening there is you're taking your glucose and you're splitting it into two. Glucose has 6 carbons in it, so when you split it in half, you get 2 molecules that each have 3 carbons in them, and we call those pyruvates. So glucose is converted to 2 pyruvates. That process requires ATP, so you have to use 2 ATP to split the glucose, which means that on the product side you get 2 ADP. Once you've spent your 2 ATP, technically you also get 2 free phosphates. And you're also going to generate ATP in that process. You actually generate 4 ATP total, which means that you need 4 ADP as a reactant and of course 4 free phosphates as well. And also you are adding electrons to 2 NAD pluses to give you 2 NADHs. This diagram over here is kind of summarizing or showing actually the glycolysis metabolic pathway. So you start with glucose on the top. You end with pyruvates on the bottom. Another word for pyruvate is pyruvic acid. So in these first 3 steps, you are basically just adding phosphates to glucose. Those phosphates actually come from ATP. So in the very first part of glycolysis is where you need to use ATP. to get phosphates to add them to that glucose. Of course, by that point, we don't technically call it glucose anymore. It has a different name. You don't need to worry about the names of all these intermediates. The next step after that is to split this molecule into two. So then we end up with two 3-carbon molecules. And at that point, the diagram here kind of splits into two to show the process that's occurring for each of those molecules, but then they actually drew both of them on each side also, so it kind of looks like there's 4, but there's only 2. So, I guess you could actually just kind of ignore one of these arrows entirely, basically. It's an odd choice on their part. But basically, you're going to have you have now split glucose, so you have these 2 molecules that each have 3 carbons in them and 1 phosphate, and that's the point at which you are going to add the electrons to NAD+, giving you NADH. That process also involves adding additional phosphates to these glucose halves. And then in the final steps of glycolysis is when you begin generating ATP, and you're ultimately going to generate 4 ATP. At the very end, you have removed all of those phosphates from the glucose halves and converted them into pyruvates. So now you have 2 pyruvates that each have 3 carbons in them. You've also used 2 ATP. You've created 2 NADHs, and you've created 4 ATP. Since you have to use 2 ATP, but you make 4 ATP, overall, you're actually making 2 ATP. So you don't actually need to remember the exact numbers for those. You don't need to know how much ATP is required in glycolysis, or how much ATP you make, or how many NADHs you make. You do need to know that you make 2 pyruvates though, and they each have 3 carbons. So generally what you need to know for glycolysis is that it requires glucose, and it requires NAD+, and it requires ATP, and that it makes 2 pyruvates, and ATP, and NADH. And that those pyruvates each have 3 carbons, glucose has 6 carbons, you split it into half to get 2 pyruvates that each have 3 carbons in them. A final point about glycolysis is that it does require the NAD+ as a reactant. So that means that if you run out of NAD+, then you cannot do more glycolysis. As you're performing glycolysis, you're taking your NAD+ and you're adding electrons to it, getting NADH. In order to get more NAD+, so you don't run out, you have to do something with that NADH. And that is going to become relevant kind of later on when we're talking about cellular respiration and particularly fermentation. The next process in cellular respiration is pyruvate oxidation. Pyruvate oxidation happens in the cytosol for prokaryotes, but in eukaryotes it happens in the mitochondrial matrix. Pyruvate oxidation uses pyruvate as a reactant. It also uses NAD+ and CoA as reactants. CoA stands for coenzyme A. A coenzyme, as you might remember, is an organic cofactor. So CoA is basically a molecule that certain enzymes need to function, and it's an organic molecule. So those are your reactants. They're going to be converted to acetyl-CoA and NADH and carbon dioxide as your products. So what's actually going on there is that you're taking your pyruvate that you generated in glycolysis and you are splitting it up. Pyruvate has 3 carbons on it, shown right here. See, each of these little balls is one of the carbons in pyruvate. In pyruvate oxidation, you are going to take one of these carbons off and add it to oxygen, which gives you carbon dioxide. The remaining 2 carbons in pyruvate are going to be added to CoA, and that gives you acetyl-CoA. These 2 carbons that were left from the pyruvate are actually an acetyl group, so when you add them to CoA, that gives you acetyl-CoA. So pyruvate is being split. 1 carbon goes to carbon dioxide, the other 2 carbons in pyruvate go to acetyl-CoA. At the same time, you are adding electrons to NAD+ to give you NADH. The carbon dioxide that you generate in this process is a waste product, so the cell is just going to get rid of it. The acetyl-CoA and the NADH are going to be useful later in cellular respiration. Oh, and then this figure here is just kind of showing where, the basic layout of a mitochondrion. So the mitochondrial matrix is the innermost part of the mitochondrion, and that is where pyruvate oxidation would be occurring. The next process in cellular respiration is the Krebs cycle, also called the citric acid cycle or the tricarboxylic acid cycle. For prokaryotes, of course, this is happening still in the cytosol. For eukaryotes, this is happening in the mitochondrial matrix, along with pyruvate oxidation. The Krebs cycle is a very complicated metabolic pathway. It's called a cycle because the chemical that you start with is also the chemical that you end up with, that is oxaloacetate. So the cycle begins with oxaloacetate and you start converting that to other molecules, but at the end of the cycle you've converted it back to oxaloacetate. The basic purpose of the Krebs cycle is to make a lot of electron carriers, or I should say to reduce a lot of electron carriers by adding electrons to them. You also make a little bit of ATP. So the reactants for Krebs cycle are acetyl-CoA and oxaloacetate, and 3 NAD pluses, and an ADP, and an FAD. The products are oxaloacetate again, also CoA, 2 CO2, 3 NADHs, and an ATP and an FADH2. Basically what's going on is you're starting with your oxaloacetate, then you have acetyl-CoA come in from pyruvate oxidation, and you're going to take these 2 carbons off of acetyl-CoA and add them to oxaloacetate. When that happens, that gives you citrate. So oxaloacetate has 4 carbons in it. The acetyl group here on acetyl-CoA has 2 carbons in it, so citrate has 6 carbons. And then you're left with just CoA by itself, so that's going to leave as a product. Then citrate is going to continue on in the Krebs cycle, being converted from one molecule to another. As you go, you are going to be adding electrons to NAD+ to give you NADH. You also will start removing carbons from citrate. So here you have 1 carbon leaving as CO2, which gives you 5 carbons left, and then you take off another carbon as CO2, and that gives you 4 carbons left. So now you have a molecule that has actually the same number of carbons in it as oxaloacetate has, but it's still different from oxaloacetate, so you actually need to do some more work to get it back to oxaloacetate. In that part of the process is where you're going to be adding a free phosphate group to ADP to give you ATP. This is actually shown as GDP and GTP in this diagram here, but is basically the same thing. And you're also going to add electrons to FAD to give you FADH2 and more electrons to NAD+ to give you more NADH, and then finally you're back at oxaloacetate, the same thing you started with. So you're generating a lot or reducing a lot of electron carriers here. You're also making a little bit of ATP, and the carbons in acetyl-CoA, these had come from pyruvate, which came from glucose. So these carbons are ultimately from glucose. Both of them are going to leave as carbon dioxide. So by the time the Krebs cycle is over, you have completely oxidized all of the carbons that were in glucose to carbon dioxide. Another point to make here is that this cycle is going to be happening twice for every glucose. Actually, same thing for pyruvate oxidation, which I think I forgot to mention. It actually is going to happen twice for every glucose. That's because in glycolysis, you take a glucose and split it into 2, so you get 2 pyruvates. Then each of those pyruvates is going to go into pyruvate oxidation and then be converted to acetyl-CoA, and each of those acetyl-CoAs will go into the Krebs cycle. So you're going to do pyruvate oxidation twice for every glucose, and you're going to do the Krebs cycle twice for every glucose. So here you have another diagram showing the Krebs cycle. Again, you're going to be starting with oxaloacetate that has 4 carbons in it. You're going to be adding 2 carbons to it that came from the acetyl group on acetyl-CoA to give you citrate. Then CoA will leave as a product. As you proceed through the Krebs cycle, you're going to be removing those carbons from citrate one by one. So once you take 1 off, then you have 5 carbons left, and that last carbon leaves as a carbon dioxide. Then you take another off and you have 4 carbons left. The last carbon leaves as, again, a carbon dioxide. Oh, here you have 4 carbons left, haha. And then as the process continues, you're adding, you're generating ATP and you're adding electrons to electron carriers. I guess a brief note on the names for this cycle. Krebs is the scientist who discovered it, so that's why it's called the Krebs cycle. It's called the citric acid cycle because of citrate that's involved in the cycle. Another name for citrate is citric acid, and it's called the tricarboxylic cycle, or the tricarboxylic acid cycle, because it has a lot of carboxylic acids, which are just acids that have a carboxyl group in them. So we'll just pause and kind of summarize what has happened so far in cellular respiration. So first we did glycolysis, then we did pyruvate oxidation or pyruvate processing, then we did the Krebs cycle or citric acid cycle. In glucose, in glycolysis, we took our glucose and converted it to 2 pyruvates. So all of the carbons in glucose went into pyruvate. Then in pyruvate oxidation or pyruvate processing, we converted those pyruvates to acetyl-CoA. Each pyruvate has 3 carbons. Each acetyl-CoA has 2 carbons that had come from glucose. The acetyl part is 2 carbons, so that means that you've lost 1 carbon in pyruvate oxidation. That carbon is lost as a carbon dioxide. Of course, pyruvate oxidation is going to run twice for every glucose, since you made 2 pyruvates from 1 glucose. So for feeding 2 pyruvates into pyruvate oxidation, you're going to make a total of 2 acetyl-CoAs and 2 carbon dioxides. That means that at that point, you started with 6 carbons in glucose. Now you've had 2 of them leave in carbon dioxide, and the remaining 4 are on acetyl-CoA and 2 different molecules of acetyl-CoA, each one having 2. Then these acetyl-CoAs are going to enter the Krebs cycle or citric acid cycle, in which the carbons in the acetyl group will be stripped off one by one and will leave as carbon dioxide. So for every acetyl-CoA, that means you're having 2 carbons leave as carbon dioxide, but since you have 2 acetyl-CoAs total for every glucose, you're going to have 4 carbons leave in carbon dioxide. At that point, out of the 6 carbons that you had in glucose, 2 of them left as carbon dioxide in pyruvate oxidation, the remaining 4 left as carbon dioxide in Krebs cycle, so now you have none left. They have all been added to carbon dioxide, which is a waste product that the cell is just going to get rid of. So that means that all of those carbons that started in glucose are now fully oxidized to carbon dioxide. They all started out having nonpolar bonds, and now in carbon dioxide, they are all involved in polar bonds in which they have kind of lost electrons to oxygen. Also to this point, we've made a little bit of ATP. We made some ATP in glycolysis. We made some more ATP in the citric acid cycle. We made a total of 2 ATPs in glycolysis and a total of, well, we make 1 ATP every time the citric acid cycle runs, but we run it twice for 1 glucose, so that means we get 2 ATP out of it. Actually, you don't need to know that it's 2 ATP specifically, just that there's a little bit of ATP that comes out of glycolysis and a little bit that comes out of the citric acid cycle. The other thing that we've generated so far that's important is our electron carriers charged up with electrons. So in each of these processes, we're making NADH. You make NADH in glycolysis and in pyruvate oxidation and in Krebs or the citric acid cycle. In your citric acid cycle, you also make FADH2. Most of the electron carriers that you're making have come out of the Krebs or out of the citric acid cycle. Those electron carriers are what's going on to the next process, which is oxidative phosphorylation. In oxidative phosphorylation, those electron carriers, or the electrons that they have at least, are going to be used to make a lot of ATP, not just a little bit. This graph is showing the energy that's available in the different products that are produced throughout glycolysis, pyruvate oxidation, and the Krebs cycle. So you start out with glucose at the beginning, and this is all considered like relative to glucose. So glucose is set to zero energy. Actually, glucose does not have 0 energy. It has plenty of energy in it. But we're really interested in comparing how other products in these processes how much energy they have as compared to glucose. So we've just arbitrarily set glucose to 0. In the glycolysis is the part of the graph that is white in background. So in the first parts of glycolysis, we are adding phosphates to glucose from ATP. We're actually using ATP. So you can see that the energy in those intermediates is going up a little bit as we add in ATP. Then we get to the latter part of glycolysis where we start taking things out. So first we're going to add some electrons to NAD+ to give us NADH. The energy from those electrons is going to go into the NADH and it leaves our intermediate. So we have a drop in energy at this point. Then we have the parts of glycolysis where we make ATP, and again, the energy, some of the energy from these intermediate molecules that came from glucose is going to be funneled into the ATP. So it's going to be leaving in the ATP. It's going to be basically captured and stored as the ATP, or as electrons on NADH. At the end, we finally make pyruvate, and you can see that it has a lot less energy than what glucose has. So all of that energy that you lost from going to glucose to pyruvate has come out in the electrons in an NADH and on your ATPs. Then you enter pyruvate oxidation, which is very short. You convert pyruvate to acetyl-CoA. You also add electrons to NADH. And you can see the energy that acetyl-CoA has is a lot less than the energy that pyruvate has. So you're again taking energy out of these intermediates. That energy is going as electrons that are added to NADH. So the energy is actually entering the NADH. Then you have your acetyl-CoA that's going to enter the Krebs cycle or the citric acid cycle. So it's also going to start losing energy. First, you're going to take those carbons and add them onto oxaloacetate, giving you citrate. Then you're going to start taking stuff out. So you're going to start adding electrons to your electron carriers, so making NADH. Each time you do that, energy leaves the intermediates. So whatever like citrate gets converted into, it has less energy because that energy left as electrons that were added onto NADH. And that happens again. You lose some more energy when you make ATP, and you continue to lose more energy when you make FADH2 and your last NADH. At the end, you get to oxaloacetate, which has very little energy compared to glucose, or compared to acetyl-CoA, of course. You know, it's lost a lot of energy. So in the Krebs cycle, you start out with, you know, this amount of energy on acetyl-CoA, and you're left with this amount of energy on oxaloacetate. All of that extra energy went into these electron carriers and into the little bit of ATP that you made. So overall, if you compare glucose to oxaloacetate, that's like 700 joules of energy, or sorry, 700 calories, there we go, 700 calories of energy that you lost going from glucose to oxaloacetate. All of that energy has been put into electron carriers and into ATP. Most of it has gone into the electron carriers. Only a little bit of energy went into ATP. And those electron carriers are what is moving on to oxidative phosphorylation. In oxidative phosphorylation, you're going to be taking the energy that you added into those electron carriers and using it to make ATP. The final process in cellular respiration is oxidative phosphorylation. Oxidative phosphorylation has two parts. First, the electron transport chain, and second, chemiosmosis. In prokaryotes, oxidative phosphorylation occurs on the cell membrane itself, and in eukaryotes, it occurs on the mitochondrial inner membrane, which is surrounding the mitochondrial matrix. So this diagram here is kind of summarizing oxidative phosphorylation. This part on the left is the electron transport chain, and then this part on the right is chemiosmosis. In the electron transport chain, basically what you're doing is using electrons from electron carriers to create a concentration gradient for protons across the membrane. And in chemiosmosis, you are using that concentration gradient for protons to make ATP. So starting with the electron transport chain, embedded in this membrane, whether it's the cell membrane in a prokaryote or the inner mitochondrial membrane in a eukaryote, in the mitochondrion of a eukaryote, which is what's shown here, you have these protein complexes embedded in the membrane. Each protein complex has the ability to carry or to hold electrons. So you're going to have electron carriers come in, like NADH and FADH2. They're going to drop their electrons off onto these protein complexes. The electrons are going to hop from one complex to the next. And at the end, they need to be added to some other molecule to take them away, and that's going to be oxygen. So at the end, you add your electrons to oxygen, and that's going to give you water. As the electrons are traveling along the electron transport chain from one protein complex to the next, the energy of their movement is being used to pump protons across the membrane, and a proton is abbreviated as an H+. So the reactants for the electron transport chain would be NADH and FADH2, also oxygen and protons. And the products are NAD+, FAD, and water. So actually, when you have these electrons and you're adding them to the oxygen, they're going to combine with protons from the solution that's around them to make water. Because water has hydrogens, not just electrons. But if you take an electron and add a proton to it, then you have a hydrogen, so that makes water. So all of those electron carriers that you charged up with electrons in glycolysis and in pyruvate oxidation and in the Krebs cycle, those electron carriers are all going to be used in oxidative phosphorylation. They're just carrying those electrons to the electron transport chain and dropping them off. So once NADH drops off its electrons, you have NAD+ again. Once FADH2 drops off its electrons, you have FAD again. Then those compounds, NAD+ and FAD, can go back to glycolysis, pyruvate oxidation, and the Krebs cycle and pick up more electrons. So you typically have 4 different protein complexes embedded in the membrane of the inner membrane of the mitochondrion for the electron transport chain. And they're just named complex 1, complex 2, complex 3, and complex 4. Each of these protein complexes is going to have multiple molecules within it that are able to actually hold and or carry electrons. Those molecules usually count as cofactors in these protein complexes. Some of them are flavins, some of them are cytochromes. One common one is ubiquinone. So it's not all the proteins in these protein complexes that are actually carrying the electrons, but it's these cofactors that are carrying them. So for instance, in this diagram, this orange Q here, that is ubiquinone. So that's going to pick up electrons from protein complex 1, from some cofactor in protein complex 1, and carry them to protein complex 3, where they'll be dropped off on another cofactor within this complex. Then you have, in green here, cytochrome C. That is going to carry electrons from complex 3 to complex 4. So you don't need to know the names of those different electron carrying cofactors within the protein complexes. Each one is going to have a different redox potential. The redox potential is the ability of a molecule to be reduced by taking on extra electrons. At the beginning of the electron transport chain, the cofactors that are carrying the electrons are going to they're going to have a lower redox potential. When they have a lower redox potential, that means that it's harder for them to accept electrons. And as you go, the redox potential is going to go up steadily. So complex 1 cofactors would have the lowest redox potential. Complex 4 cofactors would have the highest redox potential. And then oxygen actually has the highest redox potential of them all. So it is the best electron acceptor, or, you know, it's very, very good at accepting electrons and being reduced. So basically that means that when you're starting out with these electrons entering the electron transport chain, they have a lot of energy. And you don't have to be very good at accepting electrons to accept them, because, you know, they do have a lot of energy. So that's why on the complex 1 side of things, your cofactors will have a lower redox potential. Then as the electrons hop from cofactor to cofactor and move from complex to complex, they're going to start losing energy. That energy is going to be used to pump protons or hydrogen ions across the membrane, setting up a concentration gradient for protons across this membrane. So as energy is used to pump those protons, it is being lost from the electrons. The electrons get lower and lower in energy, which means it gets harder and harder for the next cofactor to accept them, which means that if it will accept them, if it's going to accept them, it needs to have a higher ability to accept them or a higher redox potential. So the redox potential is going to go up as the energy of the electrons goes down until you get to oxygen. Oxygen is the final electron acceptor, which means that when the electrons get to the end of the electron transport chain, the final molecule to accept them is going to be oxygen. Oxygen has a very high redox potential, and when the electrons get to the end of complex 4, they don't have very much energy left. So you have to have a very high redox potential in order to accept them, which oxygen does, so it can accept them. And so oxygen will pick up these electrons from complex 4, and it'll also pick up a couple protons from the solution inside the mitochondrial matrix, and that'll give you water. A final thing to note here is just that your electrons for NADH enter the electron transport chain at complex 1. Your electrons for FADH2 or from FADH2 enter the electron transport chain at complex 2, so it's a little bit later, and then both of them would be funneled to complex 3. Basically what that means is that the electrons from NADH or when NADH drops electrons off onto the electron transport chain, those electrons will travel on the chain for farther or for longer. More of their energy is going to be used to pump protons. So you're ultimately going to pump more protons using energy from electrons that came from NADH compared to energy from electrons that came from FADH2. So I guess I basically already explained everything on this slide, but just to recap, you're going to have NADH dropping off its electrons at protein complex 1 in the electron transport chain. Ubiquinone will carry them to complex 3. FADH2 drops off its electrons at complex 2 instead, and again, ubiquinone will carry them to complex 3. That means that you're going to get more protons pumped using electrons that were from NADH than what you can using electrons from FADH2, because NADH electrons travel on the electron transport chain for longer. The electrons will be carried from complex 3 to complex 4 by cytochrome C. Then once the electrons get to complex 4, they're going to be added to oxygen, which creates water. Specifically, you're going to have oxygen combining with electrons from complex 4 and with protons from the solution in the mitochondrial matrix, and that makes water, because an electron plus a proton equals a hydrogen. As these electrons are moving on the electron transport chain, they are being used to pump protons across the membrane. That is the purpose of the electron transport chain, to pump these protons and set up a concentration gradient for protons, where you have a lot of protons on one side of the membrane and very few protons on the other side of the membrane. So if this is a eukaryote, that means that you're going to end up with a lot of protons in the intermembrane space in the mitochondria, which is the little space between the outer membrane and the inner membrane of the mitochondria. In the matrix, in the innermost compartment of the mitochondria, you'll have few protons. If this were a prokaryote though, then this membrane would actually be the cell membrane, so you would be pumping protons out of the cell, and you would have only a little bit of protons inside the cell. So once again, you have your electron carriers that came from the other processes in cellular respiration, glycolysis and pyruvate oxidation and the Krebs cycle, coming into the electron transport chain and dropping off their electrons at the protein complexes of the electron transport chain. Those electrons are going to hop from protein complex to protein complex, moving down the electron transport chain. As they move, they're losing energy. That energy is being used to pump hydrogen ions across the membrane, which sets up a concentration gradient for hydrogen ions, where you have a lot of hydrogen ions, also called protons, on one side of the membrane and very few on the other side. When the electrons get to the end of the electron transport chain, they are added to oxygen, which creates water. So this graph is showing the energy associated with electrons as they move through the electron transport chain. So you start out with those electrons on NADH or on FADH2, and you end up with the electrons on oxygen. So you can see that as the electrons move through the electron transport chain, they are losing energy. These are all the different cofactors that carry the electrons throughout the electron transport chain. So for NADH, those electrons would start off on protein complex 1, and there's a couple cofactors there that they're going to hop between. Then they're added to ubiquinone, which takes them to complex 3. There's more cofactors that they hop between. Then they go to cytochrome C, which takes them to complex 4. There's a couple more cofactors that they hop between, and then finally at the end of complex 4, they're added to oxygen. For FADH2, electrons, it's the same thing basically, except that instead of being added to cofactors on protein complex 1, they enter the electron transport chain at protein complex 2. So you can see that at every step here, when the electrons move to a new cofactor, the energy that they have is going down. Just, systematically going down. When you are starting at the top of the electron transport chain and the electrons have a lot of energy, it's very easy to accept them. So these cofactors can have a low redox potential or a low ability to accept electrons. But as the electrons lose energy, in order to still accept them, you have to have a higher redox potential. So the cofactors that are at the end of the electron transport chain have a higher redox potential. And then finally, the last electron acceptor, or the terminal electron acceptor, is oxygen itself, which has a very, very high redox potential because it is so electronegative. It's really, really good at accepting electrons. So even though the electrons don't have very much energy left at that point, they can still be accepted by oxygen, in that process losing even more energy. All of this energy that the electrons are losing throughout this process is being used to pump hydrogen ions or protons across a membrane, creating a concentration gradient for protons across that membrane. So it's like you're converting the energy from the electrons into this proton concentration gradient. A final point to note here is that since oxygen has such a very high redox potential, that makes it the most efficient thing that you can use as your final electron acceptor to accept electrons that are coming off of the electron transport chain. You can theoretically use something else, but it's not going to be quite as good as at accepting electrons. Therefore, you're not going to get quite as much energy out of those electrons, and you're not going to be able to pump as many protons, and therefore you're not going to be able to make as much ATP. So oxygen is the most efficient final electron acceptor because it is so electronegative and has such a high redox potential. So we've just covered the electron transport chain, which is shown on the left part of this diagram of oxidative phosphorylation. In the electron transport chain, again, you have electron carriers coming in, dropping off their electrons, becoming oxidized back to NAD+ and FAD. These electron carriers are now going to go back to glycolysis, to pyruvate oxidation, to the Krebs cycle, and serve as reactants there. The electrons, in the meantime, are going to be traveling along the protein complexes of the electron transport chain from one complex to the next. As they move, the energy that they have is being used to pump protons, also called hydrogen ions, across this membrane, creating a concentration gradient for protons across the membrane. At the end of the electron transport chain, those electrons are added to oxygen, which creates water. So the basic point of the electron transport chain is to create this concentration gradient for protons across the membrane. Then in the second part of oxidative phosphorylation, which is chemiosmosis, we're going to use that concentration gradient for protons to finally make ATP. So chemiosmosis can be kind of broadly defined as just ions moving across a membrane, going down their concentration gradient, but it's especially used to refer to protons moving across a membrane down their concentration gradient in order to make ATP. That concentration gradient for protons across the membrane is actually called the proton motive force, because it's basically energy that's stored in this concentration gradient of protons that can be used to do something. Actually, in bacteria, there's a number of things that they can do with the proton motive force, not just make ATP. They can also do other things like, for instance, run their flagella to help them move. But in eukaryotes, it's only really used to make ATP. There is a specific enzyme, or maybe I should say it like a special enzyme, that is used in chemiosmosis to actually make ATP. It is called ATP synthase. So that is the enzyme that's illustrated here. This is ATP synthase. So this enzyme is going to allow protons to flow across the membrane. So it has a little ion channel in there for them to go across. They're going to be flowing down their concentration gradient, so that means that they're going to be moving across the membrane spontaneously. As they move, the energy of their movement is used to rotate the ATP synthase enzyme, to rotate this kind of head of it here, and as the head is rotating, it starts making ATP. It's going to start taking ADP and attaching free phosphates to it to make ATP. So we'll just go into a little bit more detail on how ATP synthase is actually working to make ATP. You have four basic parts of the ATP synthase enzyme. You have the F0 unit, or the F0 unit. Then you have the F1 unit, which is like the head of it. And connecting those two units, the F0 unit and the F1 unit, you have the shaft. And then also connecting those two units, you have the stator. The stator and the F0 unit are embedded in the membrane, and well, part of the stator is sticking out, of course, and the shaft and F1 unit are sticking into the mitochondrial matrix if this is a eukaryote, or sticking into the cytosol if this is a prokaryote. So the F0 unit is basically a protein channel that allows protons to cross through it. So these protons are going to be flowing through this channel, going down their concentration gradient and across the membrane. So this is a form of facilitated diffusion, because the protons are just diffusing across the membrane down their concentration gradient, going through the channel that the F0 unit provides. As these protons are diffusing across the membrane through the F0 unit, their movement is going to cause the shaft to start to rotate. So it's going to start spinning around. And as it spins around, the F1 unit proteins are going to have conformational changes. And remember, a conformational change is a slight change in the shape of a protein that helps it do its function. So they're going to be going through conformational changes, and those conformational changes allow them to take a free phosphate and attach it to ADP, which gives you ATP. The stator, on the other hand, is just there for structural support to make sure that the F0 unit and the F1 unit stay connected together. So it's hard to know for sure exactly how many ATP are made in chemiosmosis, funnily, but it is quite a lot. We know that you need to have 10 protons flow through the ATP synthase enzyme in order to make every one ATP. So if you're going to make one ATP using ATP synthase in chemiosmosis, you have to have pumped 10 protons across the membrane in the electron transport chain. How many protons you're pumping across the membrane is going to depend on if you're using electrons that came from NADH or if you're using electrons that came from FADH2. If you're using electrons from NADH, they enter the electron transport chain earlier and they're able to pump more protons across the membrane. So that's a bit more efficient. Our best estimates are that for every NADH, you're making about 3 ATP, and for every FADH2 that enters or drops its electrons off at the electron transport chain, you're making 2 ATP. Overall, for every glucose that enters glycolysis, you're going to make about 25 ATP in chemiosmosis. Now, you're actually making more than that, but you have to spin some because, of course, glycolysis happens in the cytosol, And then the next step is pyruvate oxidation, and that happens in the mitochondrial matrix. So you have to actually take the pyruvate from glycolysis and pump it into the mitochondria, across both membranes of the mitochondria, so it gets to the matrix. Same thing for the NADH that you make in glycolysis, that has to be pumped into the mitochondrion as well. And so that's going to cost some ATP. That reduces the efficiency of chemiosmosis. Also, some of the protons that you pump across the membrane in the electron transport chain are going to kind of leak back across the membrane rather than flowing through the ATP synthase channel, and that is also going to reduce the efficiency of chemiosmosis. So given those limitations on its efficiency, it's about 25 ATP that you make in chemiosmosis for every glucose, and that's for eukaryotes that have mitochondria. For prokaryotes, it's actually going to be different, and the number is going to vary from one species of bacteria to another, or from one species of archaea to another. One reason is that prokaryotes don't all have the same electron transport chain. They're going to have different protein complexes in there. That's going to affect how many hydrogen ions they're able to pump, or how many protons they're able to pump across the membrane. Another reason is that bacteria and other prokaryotes are able to use the proton motive force for other things as well, not just for making ATP. So for instance, they can use it to rotate their flagella and help them move through a liquid. So that also is going to reduce the amount of ATP they make if they're choosing to use their proton motive force to do something else. So if you compare that to the amount of ATP that you're making in glycolysis, which is 2, and the amount of ATP you're making in Krebs cycle, which is also 2 for every glucose, you're making a bunch of ATP in chemiosmosis and in oxidative phosphorylation for each glucose. So that is cellular respiration. So to summarize that, we have 4 processes in cellular respiration. First, you start with glycolysis. In glycolysis, you are converting glucose into 2 pyruvates. You're also generating a little bit of ATP and NADH, the electron carrier. The pyruvate that you generate in glycolysis is going to move on to pyruvate oxidation, which is not specifically labeled on this diagram, but it would be occurring right here. In pyruvate oxidation, you are converting pyruvate into acetyl-CoA. Actually, two of the carbons in pyruvate are entering acetyl-CoA, and the last carbon is going to leave as carbon dioxide. You are also, again, making NADH. Then the acetyl-CoA that you made in pyruvate oxidation is going to enter the Krebs cycle. In the Krebs cycle, the 2 carbons in the acetyl-CoA that ultimately came from glucose are going to leave as carbon dioxide. You're also going to make a little bit of ATP, and you're making a lot of NADH and some FADH2. So all of the electron carriers that you made in glycolysis and in pyruvate oxidation and in the Krebs cycle are going to enter into the electron transport chain of oxidative phosphorylation. When those electron carriers enter the electron transport chain, they're basically going to drop their electrons off at the electron transport chain. The electrons are going to hop from one protein complex to the next along the electron transport chain. The energy of their movement will be used to pump protons or hydrogen ions across the membrane, creating a concentration gradient for protons called the proton motive force. At the end of the electron transport chain, the electrons will be added to oxygen, which creates water, and the proton motive force, or that concentration gradient for protons that you created using the electron transport chain, is used in chemiosmosis to make ATP by ATP synthase. So ATP synthase will allow the protons to flow back down the concentration gradient across the membrane, down their concentration gradient, and it uses the energy of their movement to make ATP. When you include the ATP that you're making in glycolysis and the Krebs cycle along with the ATP that you make in oxidative phosphorylation, you're making about 29 ATP for every molecule of glucose through cellular respiration in a eukaryote. In prokaryotes, the number is different, and it actually varies from prokaryote to prokaryote, but it's still in that same ballpark. So here's another diagram summarizing the processes of cellular respiration. You start with glycolysis. In glycolysis, glucose is converted to pyruvate. You also make some ATP, and you add electrons to NADH, or to NAD+, which gives you NADH. The pyruvate, you're making 2 of them for every glucose. Glucose has 6 carbons. You're splitting it in half to make 2 pyruvates. Each one has 3 carbons. The pyruvate is moving on to pyruvate oxidation, where it's going to be converted to acetyl-CoA. Actually, for every pyruvate that has 3 carbons, 2 carbons are going into acetyl-CoA, and the last carbon is leaving as carbon dioxide. You're also adding electrons to NAD+, giving you NADH. The acetyl-CoA that you make in pyruvate oxidation moves on to the Krebs cycle. Each carbon that ultimately came from glucose is going to be added to carbon dioxide, or it's going to leave as carbon dioxide. At that point, all of the carbons that were in glucose have now been oxidized fully to carbon dioxide. They all started in nonpolar bonds, and they're all now involved in polar bonds, so they are oxidized. You're also making a little bit of ATP in the Krebs cycle, and you're making a lot of electron carriers. You're you're reducing a lot of electron carriers by adding electrons to them, both NADH and a little bit of FADH2. All of the electron carriers that you made in all of these processes, from glycolysis and from pyruvate oxidation and from the Krebs cycle, they are all moving on into oxidative phosphorylation and the electron transport chain. In the electron transport chain, the electron carriers are going to drop their electrons off. The electrons will hop from one protein complex to the next until they get to the end, and at the end they're added to oxygen, which makes water. As they are moving along the electron transport chain, the energy of their movement is used to pump hydrogen ions, also called protons, across the membrane, which creates a concentration gradient for protons that is called the proton motive force. In chemiosmosis, the proton motive force is used to make ATP by an enzyme called ATP synthase. So ATP synthase is going to allow those protons to flow back across the membrane, going down their concentration gradient, and it uses the energy of their movement to make ATP. And finally, we have one more diagram summarizing cellular respiration. So again, you're starting with glycolysis. This diagram is a little more detailed, so you can see that glycolysis is happening in the cytosol for a eukaryote. In glycolysis, glucose is being converted into 2 pyruvates. Each glucose has 6 carbons. There's 2 pyruvates that are made from it. Each one has 3 carbons. You're also making some ATP and some NADH in glycolysis. The pyruvate is going to be pumped into the mitochondria, into the mitochondrial matrix in a eukaryote for pyruvate oxidation. In pyruvate oxidation, the pyruvate is kind of split. 2 carbons of pyruvate are added to CoA to give you acetyl-CoA. 1 carbon leaves as carbon dioxide. And this process is, of course, going to happen twice for every glucose because there's 2 pyruvates made from every glucose. You're also making NADH in pyruvate oxidation. The acetyl-CoA that you made in pyruvate oxidation is going to move on to the Krebs cycle, also occurring in the mitochondrial matrix. In the Krebs cycle, the 2 carbons on acetyl-CoA that were ultimately from glucose are going to be leaving as carbon dioxide. Again, this Krebs cycle is going to run twice for every glucose because every glucose makes 2 pyruvates. So 2 pyruvates makes 2 acetyl-CoAs, and you're going to run the cycle twice, once for each acetyl-CoA. You're also making some ATP in the Krebs cycle, and you're charging a bunch of electron carriers with electrons. The electron carriers, NADH from glycolysis and from pyruvate oxidation, and the electron carriers from Krebs cycle, NADH and FADH2, are all moving on to the electron transport chain, which in a eukaryote is found in the inner mitochondrial membrane. So you have different protein complexes in the electron transport chain. The electron carriers would drop their electrons off at the protein complexes of the electron transport chain, and then they will hop from one protein complex to the next. As they move from one complex to the next, the energy of their movement is used to pump protons across the inner mitochondrial membrane to create a concentration gradient for protons called the proton motive force. At the end of the electron transport chain, electrons are going to be added to oxygen, which creates water. And then finally, the last part of oxidative phosphorylation is chemiosmosis, in which you have ATP synthase, allowing protons to flow across the membrane, going down their concentration gradient, and it's going to use the energy of their movement to to make ATP. Most of the ATP that you are making in cellular respiration is happening in the end, is being made by ATP synthase in oxidative phosphorylation. So the type of cellular respiration that we've just been talking about is aerobic cellular respiration. So that's the type of cellular respiration that your own cells do and that basically all animal and plant cells do. Well, not basically all, but all animal and plant cells do aerobic cellular respiration. You also have another form of cellular respiration, which is called anaerobic cellular respiration. Anaerobic cellular respiration is used by certain organisms. Most of them are prokaryotes, also a few eukaryotic, single-celled eukaryotes, but none that are big enough to see. In anaerobic cellular respiration, it's basically the same as regular or aerobic cellular respiration. The main difference is you're not using oxygen. So oxygen only comes into play at the very end in oxidative phosphorylation. It's used by the electron transport chain as the final electron acceptor. In anaerobic cellular respiration, you're basically going to use something else as the final electron acceptor instead of oxygen. There's a bunch of different things that could be used by different organisms. Some of the more common substitutes would be sulfate or nitrate, but there's also plenty of others. Always you're going to end up making less ATP than what you would be able to do with aerobic cellular respiration. And the basic reason is that oxygen is very electronegative and it has a very high redox potential. Any substitute that you would use for oxygen as the final electron acceptor is going to be less electronegative and it's going to have a lower redox potential, which means it's not going to be as good at accepting electrons, which means you're not going to be able to strip as much energy out of those electrons before you put them on the final electron acceptor. Therefore, you're not going to pump as many protons across the membrane, therefore you're not going to make as much ATP. So how much ATP you can make in anaerobic cellular respiration is actually going to depend on which electron acceptor you're using as a substitute for oxygen. It can also depend on the components of the electron transport chain. Those are usually going to be different in organisms that use anaerobic cellular respiration. The image here is an electron transport chain that's used by E. coli, which is a bacteria, so it's a type of prokaryote. So this is going to be embedded in the cell membrane of that bacteria, and it's using nitrate here as the final electron acceptor instead of oxygen. You can also notice that it's kind of shorter and more simple than the eukaryotic electron transport chain. It only has 2 protein complexes instead of 4, so it's not going to be able to pump as many protons across the membrane. But also because nitrate is not as electronegative as oxygen is, that's another reason why it would not be able to pump as many protons across the membrane, and therefore would not be able to make quite as much ATP. So cellular respiration is a really great process because you're able to make a lot of ATP that way. You can also do other things to make ATP, like you could do fermentation, which we'll talk about in a moment. But in fermentation, you're not going to make nearly as much ATP as you can in cellular respiration. But there is one major advantage that fermentation has over cellular respiration, which is kind of a pitfall of cellular respiration, which is just that in cellular respiration, if you run out of your final electron acceptor, you're kind of done. So, for you, your final electron acceptor is oxygen. For aerobic cellular respiration, it's always oxygen. For anaerobic cellular respiration, it's going to be some other molecule, like maybe sulfate or nitrate instead of oxygen. But whatever it is, you have to have a constant supply of that final electron acceptor so you can add electrons to it when they get to the end of the electron transport chain. So for you, if you run out of oxygen, then you don't have anywhere to put your electrons when they get to the end of the electron transport chain. That means that they're going to get stuck there on the electron transport chain. More electrons will come onto the electron transport chain and they'll start moving along it, but they're going to get to the end and they're going to find that there's already electrons kind of clogging up all the spots for them there, and so they're going to get stuck even further up the electron transport chain. And the chain is basically going to get blocked up or backed up with electrons. Eventually the whole thing will be full of electrons that are not moving along it, and when NADH comes to drop off more electrons, or when FADH2 comes to drop off more electrons, there's not going to be anywhere to drop them off. Everywhere that they could put them, it's already going to be full of electrons. But none of the electrons are moving because none of them can leave the electron transport chain because you don't have a final electron acceptor ready for them to move onto. If those electrons are not moving, then you're not doing anything. You're only pumping protons across the membrane if the electrons are moving on the electron transport chain. So you're not pumping protons. If you're not pumping protons, then you're not setting up your concentration gradient for protons, and you're not going to be able to do chemiosmosis to make ATP. So ATP synthase is going to shut down. Not only that, but now you don't have anywhere, like NADH and FADH2, they don't have anywhere to drop off their electrons, so they're going to be stuck as NADH and as FADH2. They need to drop off their electrons so they can go back to being NAD plus and FAD. That's because the other processes in cellular respiration have to have NAD or NAD plus or FAD. Right, so glycolysis and pyruvate oxidation have to have NAD+. The Krebs cycle has to have NAD+ and FAD. If you don't have them, then those processes cannot run. So they're going to stop as well. As soon as all of your NAD+ is converted to NADH, as soon as all of your FAD is converted to FADH2, and you don't have any way to get rid of those electrons on the electron carriers, then your other processes of cellular respiration are also going to get stuck and grind to a halt. And now you're making no ATP at all, and that's a problem. That's a big problem because if you don't have ATP, you can't run the sodium-potassium pump. And if you can't run the sodium-potassium pump, you're going to die. So this is the basic reason why you die if you don't get oxygen. Basically, if you don't have oxygen, your electron transport chain will get blocked up with electrons. You're not going to have any way to convert NADH and FADH2 back to NAD plus and FAD. You're going to stop doing cellular respiration. You're going to stop making ATP. Then you stop running the sodium-potassium pump. Then you stop manufacturing an isotonic environment for your cells to live in, and then your cells die. Of course, also some other things can happen if you don't have ATP. So, for instance, like your heart won't be able to beat. But of course, the only reason your heart needs to beat is so that it can circulate oxygen and glucose to your body for cellular respiration. But if cellular respiration isn't happening anyways because there's no oxygen, then there's really no point. Basically you'll die. So that's a major problem. The way around it is to use fermentation to get back NAD plus from NADH. So fermentation is actually going to use NADH, take the electrons off of it, and give you back NAD plus. Then if you're at least getting NAD plus and you have some glucose, you could at least do glycolysis, and you do make a little bit of ATP in glycolysis, and then you could run your sodium-potassium pump. So now we'll cover fermentation, which is a process that cells can use to get back NAD plus for glycolysis. So fermentation occurs in the cytosol for prokaryotes and for eukaryotes. There's actually a lot of different reactions that count as types of fermentation, and so the definition for fermentation can be a little vague. Different people kind of define it in different ways. But one way to define fermentation is that it is ways of using carbohydrates to make ATP without doing oxidative phosphorylation. So that means it cannot involve an electron transport chain. Fermentation is much simpler than oxidative phosphorylation and much simpler than cellular respiration. It doesn't need oxygen. And it also doesn't need something to substitute for oxygen. So anytime you're doing oxidative phosphorylation, you either need oxygen to serve as a final electron acceptor, or you need something else like sulfate or nitrate that can substitute for oxygen and act as the final electron acceptor. In fermentation, you're not using an electron transport chain, so you don't need a final electron acceptor at all. That means that it's kind of easier to do fermentation if you have fewer resources around. Fermentation actually does not make ATP directly. Instead, you make ATP in glycolysis when you're doing fermentation, and then the point of doing fermentation is to get back NAD+ so that you can do more glycolysis. Even though there's a lot of different fermentation reactions, they all have certain things in common. For instance, they all require pyruvate. You generate that pyruvate in glycolysis. They all require NADH, and they all give back NAD+. And then that NAD+ is used to do another round of glycolysis. So that's basically shown in this diagram here. The first part of the diagram is basically just glycolysis, in which you're starting with glucose and converting it to 2 pyruvates. In the process, you make a total of 2 ATP and 2 NADHs. The pyruvate and the NADH are moving on to fermentation. This particular diagram is showing lactate fermentation, which is the type of fermentation that your own cells are able to do. In this type of fermentation, pyruvate is converted into lactate, also known as lactic acid. That process requires electrons from NADH. Once the electrons are removed from NADH, then you have NAD+. NAD+ can go back to glycolysis and serve as a reactant for glycolysis. That lets you do one more round of glycolysis to get a little bit more ATP. So as I was mentioning, there's a lot of different reactions that count as fermentation reactions. Something that they all have in common is that they require pyruvate and NADH as reactants, and then they are going to produce NAD+ as a product. They also produce something else, so they're all going to convert that pyruvate to something. What exactly pyruvate is converted to is going to depend on the fermentation reaction. It could be converted to lactate, also known as lactic acid, could be converted to ethanol, which is just alcohol, could be converted to vinegar, which is acetic acid, and there's a lot of other potential products that can be made through fermentation reactions. So sometimes fermentation products are useful to us. The cell that produces them always regards those products as a waste product, so it just gets rid of it. But if you have cells that do alcohol fermentation, so they're producing ethanol as they ferment, that's useful for us in producing, beer and wine and other forms of alcohol. So some of these fermentation reactions are very useful to produce some product that, that we like to consume. Essentially, every organism is able to do fermentation reactions of some type, although a lot of organisms don't ferment very much because they would be relying on cellular respiration as a source of ATP instead. So cellular respiration lets you make a lot more ATP. In cellular respiration, you can make about 29 ATP for every molecule of glucose, whereas in fermentation, you would usually make 2 ATP for every molecule of glucose. So that's a lot less ATP if you're using fermentation compared to cellular respiration. So organisms that can do cellular respiration, whether it's aerobic or anaerobic, they would prefer to do that respiration to make a lot more ATP. But when an organism runs out of its final electron acceptor, then it needs to be able to switch to fermentation so that it doesn't just die, and it can still continue making some ATP in glycolysis. So this happens in your own cells, most commonly in your muscle cells. If you're working out and you run through your supply of oxygen, then the cells need to do something else to create ATP, and they're going to switch to fermentation. And your cells can do lactate fermentation. So at that point, your muscles would begin doing fermentation to produce lactic acid as a waste product. Just in order to make a little bit more ATP for themselves. So again, here's our summary diagram showing cellular respiration and fermentation. Both processes begin with glycolysis, in which glucose is converted to 2 pyruvates. Glycolysis also produces a little bit of ATP, and it adds electrons to NAD+, giving you NADH. If you're doing respiration, the pyruvate produced in glycolysis moves on to pyruvate oxidation, in which one carbon from the pyruvate is split off and leaves as carbon dioxide, and the other 2 carbons are added to CoA to give you acetyl-CoA. Pyruvate oxidation also produces NADH, even though that isn't shown on this diagram. Acetyl-CoA would be moving on to the next process, which is the Krebs cycle. In the Krebs cycle, these two carbons in the acetyl group on acetyl-CoA, these two carbons originally came from glucose, they're going to be stripped off and they will leave as carbon dioxide. At that point, every carbon from glucose has now been fully oxidized to carbon dioxide. The Krebs cycle is also going to produce a little bit of ATP and a lot of NADH and some FADH2 as well. So it's producing mostly electron carriers, and that's the main point of doing the Krebs cycle, is to add electrons to electron carriers. Then all of those electron carriers from glycolysis and from pyruvate oxidation and from the Krebs cycle are going to move on to oxidative phosphorylation, and they will drop their electrons off at the electron transport chain. The electrons are going to hop from one protein complex to the next on the electron transport chain. As they move, they're losing energy. The energy is being used to pump protons across the membrane to create a concentration gradient for protons called the proton motive force. At the end of the electron transport chain, the electrons will be added to the final electron acceptor, which is usually oxygen. For aerobic respiration, it would always be oxygen. That creates water. If you're doing anaerobic respiration, then you're using something else instead of oxygen, and you would be producing something else instead of water. Then ATP synthase is going to use the proton motive force that you generated using the electron transport chain to make ATP. So that process is called chemiosmosis. ATP synthase is going to allow protons to flow back across the membrane, going down their concentration gradient, and it's going to use the energy of their movement to make ATP. This way you can make a lot of ATP. If you're doing cellular respiration, you make a lot. Most of what you make is made in oxidative phosphorylation by ATP synthase. But if you run out of the final electron acceptor, then the whole process will shut down. The electron transport chain will get backed up with electrons, and you won't be able to remove electrons from electron carriers, which means you're going to be stuck with NADH, and you won't be able to get back NAD+. So all of these processes that require NAD+ are going to shut down, and that's all of them. Then you're going to not be making any ATP at all. So when that happens to cells, they're going to switch to doing fermentation. In fermentation, you again are starting with glycolysis, producing 2 pyruvates from glucose, also producing some ATP, or a little ATP, and a little NADH. The NADH and the pyruvate are going to move on to the fermentation reaction. There's lots of different fermentation reactions that are possible. The one that your cells do would convert pyruvate into lactic acid or lactate. In that process, it's going to use the electrons from NADH, which gives you back NAD+. Then NAD+ can feed back into glycolysis and let you do another round of glycolysis for that little bit of ATP that you can make in glycolysis. So far in this chapter, we've been talking about ways that cells can use glucose to make energy in catabolism. But cells can also use other molecules other than glucose to make energy, which we had mentioned. The cells can use other sugars, or they can use fats or proteins. So we'll just really briefly go over some of the ways that cells can do that. Essentially, anytime a cell is going to use some other molecule, like a different sugar or a protein or a fat, as a source of energy for making ATP, first they need to modify that molecule so that it resembles some intermediate that is already existing in cellular respiration. So if you're doing glycolysis and you're converting glucose to pyruvate, that's like a 10-step process. So you're not converting glucose directly to pyruvate, but you're doing small changes to the glucose each time to make it look a little bit more like pyruvate until you get to pyruvate at the end. But after each modification, you technically don't have glucose anymore. You have some other molecule that has its own name, and that would be an intermediate. So if you can make some other molecule look like one of those intermediates, then it can just enter cellular respiration and be used in the normal pathways to make ATP. So this is the basic strategy that cells use to get ATP out of molecules like proteins or fats. This diagram at the bottom is summarizing how proteins and fats and sugars enter cellular respiration pathways. So if you start with a sugar that's not glucose, then you can usually just convert it to glucose, or if not glucose, then some other intermediate in glycolysis that's similar to glucose, and then it can just enter glycolysis and proceed from there. If you're starting with a fat and you're trying to get energy from that fat, first you're going to break the fat into its constituent parts. Fats consist of 3 fatty acids and a glycerol molecule, so you would break them apart into the glycerol and free fatty acids. The glycerol actually is basically a 3-carbon sugar, so it can just enter glycolysis. It already looks very much like an intermediate in glycolysis, so you just modify it a little bit and then it enters glycolysis. The fatty acids are going to be chunked up into 2-carbon chunks, and those 2-carbon chunks will be added to CoA as acetyl groups to make acetyl-CoA. Then acetyl-CoA can enter the Krebs cycle. If you're trying to catabolize proteins, then you need to chop them up into their individual amino acids that make them up, and you would remove the amino group from the amino acids, and then the rest of the amino acid can enter either pyruvate oxidation or the Krebs cycle, depending on which amino acid it is. So for lipids, to go into that in a little more detail, specifically this would be fats and potentially phospholipids that could be metabolized or catabolized in this way, not all lipids. But yeah, so for fats specifically and maybe phospholipids, they can be catabolized for prokaryotes in the cytosol, or for eukaryotes in the mitochondrial matrix. First, you need to break the fat apart into its component parts. So you'll have an enzyme called a lipase that is actually going to split the glycerol off from the fatty acid part of the fat. Glycerol can then enter glycolysis, and then you have these free fatty acids. Each fatty acid is a long chain of carbon atoms with a carboxyl group on it somewhere. Fatty acids are going to enter the process of beta oxidation. In beta oxidation, the fatty acid is chopped up into 2-carbon chunks. Each 2-carbon chunk ends up as an acetyl group that is attached to acetyl-CoA. So this diagram is kind of summarizing that process. You start with a fat at the top. You have your 3 fatty acids, each attached to a glycerol. Well, all of them attached to the same glycerol. A lipase enzyme would come and break the glycerol off, and then you would have these individual free fatty acids left over. The glycerol is going to enter glycolysis. The fatty acids are going to move on to beta oxidation. In beta oxidation, you take a fatty acid and you break off a 2-carbon chunk off of it and attach that chunk to CoA to give you acetyl-CoA. This process also actually adds electrons to electron carriers, so it also generates FADH2 and NADH. So the acetyl-CoA can go on to the Krebs cycle and be used to generate even more electron carriers, and the FADH2 and NADH that you generate in beta oxidation can go directly to the electron transport chain and drop off their electrons there. So in this way, you're really able to get a lot of energy out of a fat. First, you get energy from the glycerol, and then the free fatty acids that it has can be chopped up to make a bunch of different acetyl-CoA molecules and charge up a bunch of electron carriers with electrons. So you're really going to be able to make a lot of ATP out of that. But since beta oxidation does require NAD plus and FAD, you have to be able to turn over the electron carriers. You have to be able to get electrons off of NADH and FADH2 once you've generated them, so that the FAD and the NAD plus can go back to pick up more electrons for more beta oxidation. You can also use proteins as a source of energy. In that case, first you have to break the protein up into its individual amino acids. A protease enzyme will do that. That can either happen outside the cell for a prokaryote, or if you're a eukaryote that has a lysosome, then you can just do that inside of the lysosome. Then you're going to transport those amino acids either into the cell or outside of the lysosome into the general cytosol of the cell. And the first step, once you get the amino acid kind of in the cell, is to break off the amino group, and that's called deamination. The amino group has a couple of different fates. You could actually use it to make a new amino acid of a different type, or it's a waste product. that amine group from the amino acids is going to form urea. There's also other nitrogenous waste compounds that it could form. Urea is one of the main wastes that's found in your urine. That's actually the waste that urine is named after. So generally for your cells, when an amine group is broken off of an amino acid in deamination, that amine group would go to form a waste product, urea. The rest of the amino acid, which is basically the carboxyl group and the alpha carbon and the side chain, is going to go into some cellular respiration process. Where it enters cellular respiration depends on what amino acid it is and what its side chain is. So the different amino acids that have different side chains will look like different intermediates for cellular respiration. Some of them look more like pyruvate, and they're going to enter pyruvate oxidation. Some of them look like acetyl-CoA or some other intermediate from the Krebs cycle, and so those are going to enter the Krebs cycle. That's kind of shown in this diagram here. Here you have glucose, then after glycolysis you convert that to pyruvate, then in pyruvate oxidation you convert pyruvate to acetyl-CoA, and then this big circle is the Krebs cycle. In these boxes you have the names of different amino acids, and that indicates where they would enter. So for these amino acids like alanine and cysteine, once you break the amine group off of them, they look like pyruvate, and so they're going to enter pyruvate oxidation. You also have a bunch of amino acids, including tryptophan and phenylalanine that look like acetyl-CoA once you take their amino group off, and so they're going to enter the Krebs cycle as acetyl-CoA. Also, you have all of these other amino acids that look like different intermediates from the Krebs cycle, so they're going to enter the Krebs cycle at different points. So for microbes, which would be microorganisms, a lot of which are prokaryotes, they're just organisms that are too small to see with your eye. A lot of them will, do protein catabolism pretty regularly as a normal aspect of their metabolism, but for organisms that are larger, like you don't really do protein catabolism unless you really have nothing else available to make energy off of. So when your body is going to take in a protein and break it down, it's actually going to just usually reuse those amino acids, so it's not going to deaminate them at all. It's just going to turn around and recycle them to build a new protein. The only time that you will have a significant amount of proteins and amino acids broken down and used in catabolism to get ATP is if you have no other energy source, which would basically mean that you are otherwise starving, or just eating a diet exclusively of protein. The last thing we're going to talk about in this chapter is anabolism, really briefly and generally, just because a lot of those anabolic pathways actually touch on cellular respiration as well. So we already know that cellular respiration can be used for catabolism for a lot of different molecules other than glucose. You basically try to convert that molecule into an intermediate for glycolysis, and then it enters intermediate for glycolysis or for some other process in cellular respiration, and then it's going to enter, glycolysis or whatever process the intermediate that it has been converted to, belongs to. But you can also actually kind of run those processes or those pathways in cellular respiration in reverse to get larger molecules out of them, starting from smaller molecules. So probably the main example would be glycolysis, which just takes glucose and converts it to 2 pyruvates. You can run this pathway backwards and then start with pyruvate and get back glucose. That's called gluconeogenesis. So essentially any of these pathways in cellular respiration can be run backwards. So if you have citrate in the citric acid cycle, you could actually convert that to oxaloacetate and acetyl-CoA. If you have acetyl-CoA, you can actually convert that to pyruvate. If you have pyruvate, you can convert that to glucose. For fats, we had said that when you're going to catabolize fats, you're going to break the fat apart into glycerol and fatty acids. The glycerol will enter glycolysis, and the fatty acids will be converted to acetyl-CoA and enter the Krebs cycle. But you can actually do the reverse process as well. So glycerol is going to be converted to one of the intermediates for glycolysis. So if you're running glycolysis and you get to that intermediate, you could actually just siphon that away from glycolysis and convert it to glycerol. And now you can use that to build a fat. If you have acetyl-CoA, you can feed that into the Krebs cycle to charge up electron carriers, or you could siphon that acetyl-CoA away and use it to build fatty acids. Then you could combine those fatty acids with the glycerol and make a fat. So most of these pathways that are involved in catabolism can actually be turned around and run the other direction in order to build some larger molecule, which would be anabolism. The only real exception to that is oxidative phosphorylation itself. The electron transport chain cannot be run in reverse, so it's impossible to reverse oxidative phosphorylation for anabolic purposes. Oxidative phosphorylation can only run one direction, but all of these other processes, glycolysis, pyruvate oxidation, and the Krebs cycle, they can go forwards for catabolism or they can go backwards for anabolism. So the only anabolic pathway that we're actually going to mention by name is gluconeogenesis. Gluconeogenesis is the process of making glucose from things that are not carbohydrates. So that it could include amino acids or fatty acids or glycerol. Essentially, it could include any intermediate from pyruvate oxidation or from the Krebs cycle or even from glycolysis. Gluconeogenesis basically is just the reverse of glycolysis. It's just glycolysis running backwards. There's a couple of steps that differ between gluconeogenesis and glycolysis, but mostly it's just glycolysis backwards. The other main difference is that instead of generating ATP, it requires ATP. And it also doesn't add any electrons to electron carriers. So this diagram here is kind of summarizing gluconeogenesis, which is the process shown in the box here, and different things that can enter gluconeogenesis. So basically you're starting with pyruvate in gluconeogenesis and converting it to glucose. Glucose could then be used to build larger carbohydrates in the cell, but also if you have acetyl-CoA, you can just reverse pyruvate oxidation to convert that into pyruvate, and then that could enter gluconeogenesis. If you have fatty acids, you can chop those up to make acetyl-CoA. Then acetyl-CoA can enter gluconeogenesis if you convert it to pyruvate. If you have intermediates, actually the Calvin-Benson cycle is from photosynthesis, but if you have intermediates from this cycle here, those can enter gluconeogenesis as well. So, in our next chapter, we're going to cover what this cycle is exactly. But if you have proteins, you have amino acids that you got from chopping up proteins, some of those can enter gluconeogenesis. Glycerol also can enter gluconeogenesis. So there's a lot of different molecules that can become substrates for gluconeogenesis. All you have to do is modify them so they look like one of these intermediates in gluconeogenesis, and then they can enter the pathway. So since gluconeogenesis is basically glycolysis backwards, that also means that most of these molecules can enter glycolysis and go forwards. So if we just take glycerol as an example, once you produce glycerol by breaking down a fat, to get free fatty acids and glycerol. Glycerol can either enter glycolysis and be used to make pyruvate and ATP, or it can enter gluconeogenesis and be used to make glucose. Which way it goes just depends on what the cell needs at that time. So you're actually going to have different enzymes catalyzing the reactions for glycolysis versus gluconeogenesis. So if the cell has made the glycolysis enzymes and you get some glycerol available, then you're going to be using that glycerol in glycolysis to make ATP and pyruvate. On the other hand, if the cell has the enzymes for gluconeogenesis made, then you make some glycerol and that's going to enter gluconeogenesis to make glucose. So if the cell has a greater requirement for ATP at the moment, then it's probably going to have glycolysis active, and glycerol will be used in glycolysis to make ATP and pyruvate. But if the cell has plenty of ATP at the moment and it's trying to build some cellular structures instead, then it's more likely that you would have gluconeogenesis active, and glycerol will be used in gluconeogenesis to make glucose. So we have special names for these metabolic pathways like glycolysis and gluconeogenesis that can go one way or the opposite way, that can run forwards or backwards. Those are called amphibolic pathways. An amphibolic metabolic pathway is one that could go forwards for catabolism, or it could also go backwards for anabolism. So most of these pathways we've been talking about in cellular respiration are amphibolic, and they can go back both ways. The only real exception is oxidative phosphorylation that can only go one way for catabolism. It cannot go backwards for anabolism. A cell needs to be able to control which way those pathways are proceeding, whether they're doing catabolism or whether they're doing anabolism. In general, cells are going to control that by controlling the enzymes that are available to catalyze the reactions necessary for that pathway. So glycolysis requires one set of enzymes, gluconeogenesis requires a different set of enzymes. If you want to do glycolysis, you need to make sure you have all the glycolysis enzymes made. If you want to do gluconeogenesis, then you need to turn those glycolysis enzymes off and make some gluconeogenesis enzymes instead. So cells are able to regulate how much of an enzyme they have, or where that enzyme is in the cell, or whether the enzyme is active or not. And by regulating those enzymes, that's how they're able to control whether their amphibolic pathways are proceeding forwards for catabolism or proceeding backwards for anabolism. And cells can always switch it up depending on what they need most of at the time, whether it's larger molecules for building stuff in the cell or whether it's ATP. So this diagram is kind of summarizing cellular respiration in the middle here, and how different molecules enter cellular respiration for catabolism or exit for anabolism. So first, if you're doing cellular respiration, you have glycolysis, then you have pyruvate oxidation, converting pyruvate to acetyl-CoA, then acetyl-CoA enters the Krebs cycle, then you have the electron transport chain and oxidative phosphorylation. If you're running glycolysis backwards, then that is gluconeogenesis. So all these pathways can be run backwards as well. Pyruvate oxidation can go backwards. Krebs cycle doesn't really need to go backwards specifically because it's a cycle, so it always goes through all of the intermediates no matter what. If you have lipids, you can, or specifically if you have fats, you can break those into glycerol and free fatty acids. Glycerol can enter glycolysis as an intermediate and be used for energy. The fatty acids in beta oxidation can be broken down into 2 carbon chunks that are added to CoA to give acetyl-CoA. That can enter Krebs cycle for energy. But on the other hand, if you needed to build fats, then you could take acetyl-CoA out of the Krebs cycle and use it to make fatty acids, and you could take this intermediate out of glycolysis and use it to build glycerol. And then, you could add the glycerol to those fatty acids to get back a fat. For proteins, it's a similar process. You would first chop those proteins up into amino acids. You're going to remove the amine group from the amino acids in deamination. Then the rest of the amino acid can enter some cellular respiration process. Some of them enter pyruvate oxidation. The rest of them enter Krebs cycle. But if you needed to build amino acids instead, then once you get pyruvate, you can use pyruvate to build certain amino acids if you also add an amine group to them. And acetyl-CoA and other intermediates from the Krebs cycle can also be used to build other amino acids after you've added an amine group to them. Then you can link those amino acids together to make a protein.